we are going to see the proteins. What are the proteins? Means it is a polymer of amino acid. Carbohydrates are polymer of glucose, but protein is the polymer of amino acid. The next one, classification of amino acid, properties of amino acid. Next, classification and structure and importance of protein, enzyme and liquid. These are the topics we are covered in this video. Rest of the lesson will be.
Protein, it is the main functional unit of all the living system, and they are involved in each and every function of the cell, including the respiration. And also it is the most abundant biomolecules in all living organism. So that the protein, the word is derived from the Greek word proteus. What is the meaning of proteus? Means it is a primary, or it holds the first place. The protein is the only biomolecule, it holds the first place, so that the name is given as protein. And protein, it is a polymer of alpha amino acid. Amino acid, you see the structure given there. Carbon valency we know 4. So around the carbon, the central carbon, there is 4 bond. One bond is occupied by R. R means alkyl groups. One bond occupied by hydrogen. Another bond occupied by acid. Another bond occupied by NH2. NH2 means amine. So NH2 and then acid, both are functional group. So nearer the functional group, whatever carbon present, that carbon is called as alpha carbon. Here the central carbon is nearer to acid as well as amine. So this carbon is called as alpha carbon. The protein, it is made up of alpha amino acid. Amino acid means it contains amine as well as acid, so that it is called as amino acid. And then alpha amino acid means the functional group, both the amine and acid groups, nearer one carbon is present, that carbon is called as alpha carbon. That amino acid is called as alpha amino acid. So the protein molecules, they are made up of these type of alpha amino acid. That amino acid, they are containing both the amino group as well as carboxylic acid groups. Protein molecule, they are containing 20 alpha amino acid. Commonly it present. In writing that amino acid, it given as trivial name, a 3-letter code and a 1-letter code. In writing the sequence of protein, protein means it is made up of alpha amino acid. So the sequence of protein, it is written either 1-letter code or 3-letter code. Amino acids are classified. They are based on the R groups. What is R group? Means alkyl group. It is known as side chain. Then another classification is acidic, basic, neutral amino acid. That amino acid is acid character is more, means that is called as acidic amino acid. Sometimes base character is more, means it is called as basic amino acid. Both the character is having, means that is called as neutral amino acid. Then another classification is polar and non-polar. Polar means it is hydrophilic. Non-polar means hydrophobic amino acid. And third one, essential and non-essential amino acid, based on the ability to synthesis the amino acid by the human body. So which one synthesis by the human body, that is called as non-essential amino acid. Which one it derived from the diet, that amino acid are called as essential amino acid. There are 10 essential amino acid. They are memorized by the mnemonic called PVT Tim Hall. We will see the type of amino acid later. The 3-letter code, what is the 3-letter code? We will discuss with the subsequent slide. And fourth type of amino acid is other than 20 alpha amino acid. That amino acid is called as non-protein amino acid. Usually protein is made up of only 20 alpha amino acid. So other than that 20 alpha amino acid is called as non-protein amino acid. For example, ornithine, citrulline, these are all the components we derived from the urea cycle. What is the urea cycle means? In this urea cycle, ammonia is converted into urea. I am going to explain alpha amino acid, how it is changed as a zwitterion. Alpha amino acid, COOH is a acid, NH2 is a base. According to Lowry-Brons's theory, acid it gives proton. So that COOH it gives proton and become a COO minus. Then base means it accepts the proton. So that the NH2 it accepts the H plus from the COOH and it becomes NH3 plus. This one is called as zwitterion. Now you concentrate on number 1. Number 1 structure is it contains COO minus NH3 plus. So plus and minus both the one having, so the net charge is 0. Why the net charge is 0 means plus 1 minus 1. If you add means we get the amount 0. So net charge 0. This point is called as isoelectric point and the pH value is neutral here. Now you concentrate on second diagram. The second one we get when the zwitterion is added with the H plus. H plus means when we are going to add acid. Then the H plus is attracted by COO minus. I indicated in blue color that blue color COO minus attract the H plus, so that the COO minus now change the COOH in the second diagram, but the NH3 plus is acidic. So in this one that is called as acidic pH because it is acid we are adding. Acid adding means H plus indicate that it is a acid one. Here the net charge is plus 1 because the NH3 plus only there is no minus. Now in the second diagram to first diagram, if you want to change means you have to add OH minus, that is base. That OH minus attract the H from the COOH, so that again the COOH become a COO minus, so that we get the first diagram now, that is zwitterion. Now we are going to move the first diagram to third diagram by adding OH minus. OH minus means base. That OH minus it attract the H plus from the NH3 plus, so that the NH3 plus becomes NH2 in the third diagram. In this point that is called as alkaline pH. The net charge is here minus 1 because the COO only having minus 1, but the amine there is no plus character, so that the net charge is minus 1. Once again if you add acid means that acid is attracted by base, so that it becomes zwitterion, that is diagram 1. Amino acids are crystalline solid. They are colorless and also they are water soluble solid. They have both carboxylic acid group and amino group. Based on the pH of the solution, sometimes the acid it loses proton and become negatively charged. Sometimes based on the pH, the amino group it accept the proton and it become positively charged. At specific pH, that is the net charge is 0, that point is called as neutral isoelectric point, and in this point the amino acid is act as a neutral because it contains both the positive character as well as negative character. Above the pH value, the amino acid it possesses negatively charged, and below the pH value it possesses positive charged. In aqueous solution, what happened, the amino acid it transferred one of the proton from the carboxyl acid group to amino group, and amino group it accept the positive proton from the acid and become as a positive charge, and acid it become as a negative charge. So in this point the amino acid behave as a amphoteric character and also neutral one. That ion is called as deuter ions. So deuter ion means in neutral pH value the amino acid become as a deuter ion. General structure of amino acid is around the central carbon it has one H and one side COH, one side NH2 and one side R, that is alkyl group. Based on the alkyl group there are different type of amino acid. The first amino acid is glycine. The R is replaced with the hydrogen. Here around the carbon there is two same group, that is two hydrogen is there, and two different groups, that is acid and amine. So which carbon possesses different type of group, that carbon is called as chiral carbon. But here in glycine it is not a chiral one because it has two same group, that is two hydrogen. so it not act as a optically active compound. But in another one, alanine, that in place of R we are going to replace CH3, that is methyl group. Here around the carbon there is different group are having one side CH3, one side H, and one side COOH, one side NH2. That compound is called as alanine, and symbol is capital A, and three letter code is ALA. The next one, instead of R in general structure of alpha amino acid, we are going to place isopropyl. Isopropyl means CH3, CH, CH3. That amino acid is called as valine, symbol is V, and three letter code is VAL. In this alanine and valine, around the carbon there are different type of groups are present, so they are called as chiral carbon. Rest of the amino acids have, except the glycine, rest of the amino acid, all the amino acid, they have the chiral carbon, so they act as a optically active compound. But glycine alone, it is not having the chiral carbon, so it is not act as a optically active. And there are two other forms configuration, D, capital L amino acid. That L amino acid only most of the living organism it having for the synthesizing proteins. But the presence of D alpha amino acid, it is rarely present in the organism. It present in certain organism alone. In this slide we are going to see the types of amino acid. That amino acid here given as a deuterion format ma. Just we are going to concentrate only on the R. R means side group, alkyl group. Instead of R, H is added, means that is glycine. G is a symbol and CLY is a three letter code. It is come under the category of non-polar side chain. Non-polar means there is no positive, negative, any symbol is not present in the side chain. That is called as non-polar side chain. First of all, we are going to see the non-polar side chain, glycine. Next one, alanine. Alanine means instead of R, CH3 is added. Valine is instead of R, isopropyl. Leucine, Luucine. Leucine, symbol is L. Three letter code LEU. Instead of R, we are going to add isobutyl. Isobutyl is added, means that is called as leucine. Next one, isoleucine means instead of R, secondary butyl is added, means that is called as isoleucine. Why it is called as isoleucine means it is in the iso position that amino acid is added to the butane. So it is called as isoleucine. Symbol is small l and three letter code is LIE. The next one, methionine. Methionine, instead of R we are going to add CH2, S, CH3. Symbol is capital M and MET is the three letter code. Next one, in aniline, CH3, instead of CH3 one hydrogen is removed. Instead of that hydrogen, phenyl group is added, means that is called as phenyl aniline. Symbol capital F, three letter code PHE. Next, tryptophan, symbol W, instead of R, indole ring as a side chain, that is tryptophan. Three letter code is TRP. The next one, proline. Proline, it contains 3 carbon as a cyclic one, symbol P, and three letter code is PRO. The next one, polar side chain, instead of R, that CH2OH is added, means it is called as serine, symbol S, and three letter code SER. The next one, 309, symbol is capital T, three letter code THR, instead of R, CHOH, CH3 is added. Next is cysteine, symbol C, three letter code CYS, instead of R, CH2, SH is added. Next, tyrosine, symbol Y, TYR is a three letter code, instead of R, CH2, C6H4, OH is added. Next, asparagine, symbol is capital N, three letter code ASN, instead of R, CH2, C double bond O, NH2, that is ethanamide is added. Glutamine, symbol Q, GLN is a three letter code, instead of R, propanamide is added. These are come under the category of from serine to glutamine come under the category of polar side chain because they are having the polar groups in the side chain. Next, the electrically charged side chain means they having asparagate symbol DAsP. In asparagine means it has amide ma, then asparagate it has acid, that is acetic acid. The next one, glutamate means it has propanoic acid. That propanoic acid it having the acid group, it is gives the H, because acid always gives H plus. The same way here the H plus is given away. The next one, lysine. Lysine instead of R, CH2 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 and then NH3 plus, that is called as lysine. Symbol is capital A, 3-letter code LYS. The next one, arginine, symbol capital R and 3-letter code ARG. Instead of R, CH2 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 NH, C double bond plus NH2 and one more NH2. The next one, histamine. It contains inside imidazole ring, that is histidine. Symbol is capital H, 3-letter code is HIS. See the glycine. When glycine it is added with the alanine, we remove water. The glycine, the acid group gives OH and amine group H we are going to remove, so that we get, afterward we are adding, we get NH2 CH2 CO NH CH3 COOH. In this one the name is glycyl aniline. The first one is glycine aniline, so that glycyl aniline. In that one, 2 amino acids are added, so we get dipeptide. The CONH is called as peptide bond. It is called as, it is amide linkage. Bond name is peptide bond. We are going to add 2 amino acids, so we get dipeptide. Suppose we add 3 amino acid means we get tripeptide, 4 amino acid means tetrapeptide. Like this we can say. If more number of amino acids are added, they are called as polypeptide. Large number of amino acids are added means we get the protein structure. Then amino acid end, amino end is having means the peptide bond amino end is having means it is called as N terminal or amino terminal, while the carboxyl end is called as C terminal or carboxy terminal. Usually the protein it is start from N terminal to C terminal. All other side chain except the side chain, the all other main group is called as backbone of the polypeptide. So that the amino acids they are linked by covalently with the help of peptide bonds. Proteins are classified based on their structure. One is fibrous, another one globular. Fibrous it is a linear molecule. See the structure, it is a linear one. And globular protein they are spherical. Overall structure is spherical in shape. And fibrous protein they are insoluble in water, but in globular protein they are soluble in water. In fibrous protein they are linked by, held together by disulfide bridge and weak intermolecular hydrogen bond. Example for fibrous protein are keratin, collagen. Globular protein example myoglobin, insulin. What are proteins? Proteins are polymer of amino acid. So amino acid they are linked together by peptide bond. The three-dimensional structure of protein we are get from the sequence of amino acids. So the three-dimensional structure depends only on the residue. What is residue means sequence of amino acid. There are 4 levels in the structure of protein. One is primary, secondary, tertiary and quaternary. Let's see one by one. First of all, primary structure. In this primary structure, the amino acid they are connected to the peptide bond. Then we get the polypeptide chain, that is called as primary structure of protein. If we have the knowledge in the amino acid, otherwise if we change the amino acid sequence, the alter the overall structure of protein and also the function of protein, so that we have the knowledge about that the sequence of amino acid. It is a main important in the primary structure of protein. We get the backbone structure from primary structure. Now we get the secondary structure with the help of hydrogen bond. That hydrogen bond, it formed between the carbonyl oxygen and neighboring amine hydrogen of the main chain. We get two type of secondary structure. One is alpha helix, another one beta strand. Strand means sheet. The two most common structure it is formed by proteins. First one, alpha helix, it is coiled structure. We get the right-handed helical structure. That structure, it is stabilized by hydrogen bond formed by one carbonyl oxygen of one amino acid that we have to taken as a nth residue. Afterward, the amino hydrogen of the fifth residue. So between these two hydrogen, that hydrogen bond is formed. That hydrogen bond only is responsible for this right-handed helical structure. Then the side chain we are getting amino acid. The side chain is we are getting from alkyl groups. That alkyl groups are extended outside of this helical. You see the structure alpha helical, that red color, brown color. That brown color R is called as side chain. That side chain only extended outside of the helical. Then each turn of alpha helical, they contain how many residue? Means 3.6 residue. What is the meaning of residue? It is sequence of amino acid only called as residue, and the length of the residue is the sequence of amino acid is 5.4 Armstrong degree. Then the twist of the helical structure, it is given by one amino acid that is called as proline. That proline structure is given there. It is a rigid cyclic structure. Because of this cyclic structure only, it gives a curl, king, K-I-N-K. King means curl or twist to that narrow amino acid sequence. So that only the narrow amino acid sequence that is turned into helical structure. So that the proline amino acid is called as helix breaker. It gives a break to the linear structure. So that the proline is called as helix breaker. The next one, the second structure is beta strand. This is seed structure. You see that the white color is one of the strand, and then light blue color is another strand. Between these two strands, there is hydrogen bond is formed between the carbonyl group of one such strand and amino group of another adjacent strand. That is only give the stabilization of these strand, so that it gives the beta sheet. Tertiary structure, this structure we are getting from the secondary structure. In secondary structure we have the elements alpha helix, beta sheet. They are further fold and it form the three-dimensional arrangement. That arrangement is called as tertiary structure. In this tertiary structure, they are stabilized by some interaction between the side chain of the amino acid. What are all the interaction means? One is disulfide bridge. They are formed by cysteine residue. Another one, electrostatic interaction, hydrophobic interaction, hydrogen bond interaction, and van der Waals interaction. So because of this interaction, the structure of tertiary structure of proteins are stabilized. The next one, quaternary structure. The quaternary structure we are getting from the more than one polypeptide chain. That polypeptide chain, the more than one polypeptide chain, they are interact with each other and they form multimeric structure. That structure is called as quaternary structure. You see that hemoglobin, in the structure, that hemoglobin, it contains 4 different color. Each color indicate that it is a polypeptide. So in hemoglobin, how many polypeptide is there? 4 polypeptide chain. And in DNA polymerase, it is the enzyme. It makes the copies of DNA, and this enzyme, it has 10 polypeptide chain. So what are all the interaction we studied in the tertiary structure? That interaction is a responsible for the stabilization of tertiary structure also. Sorry, quaternary structure also. There are so many chemical reaction undergo in our body. That chemical reactions are catalyzed by enzyme. What are all enzyme means? They are made up of proteins. So protein act as a catalyst for the biochemical reaction. The next one, for example, keratin, collagen, they act as a structural backbone. Keratin, collagen are protein. The next one, protein, they act as a transporter. Molecule like hemoglobin, organelles like kinesins in the cell and control the movement of molecule in and out of the cell, so that we can say that protein as a transporters. Then antibodies also proteins only. They help to body to fight various diseases. So you may call the proteins as a fighter. The next one, proteins you may call as a messenger. For example, insulin and glucogen, they are proteins. They control the glucose level in the blood. Whenever the glucose level is increases, immediately they send the messages, then only they coordinate many functions. So that protein may called as a messenger. And then the protein is called as a receptor. They detect the presence of certain signal molecule and activate the proper response. So that protein may act as a receptor. And also protein, it acts as a storage, or it store metals like iron, that is ferrin. For normal functioning of our body, we need energy. How can we get the energy? We get the energy from food. From the food we cannot get the energy directly. First of all, the food should be digested. So digestion of food, afterward we harvesting the energy from them. That energy we used for synthesis of necessary molecule. So these are all the functions in they are happen in our body. That functions, they are catalyzed by some special proteins. That proteins are called as enzyme. In our body, these reactions are occurred in the rate in the order of 10 power 5 with the help of this enzyme. It is highly specific. For example, we are going to see three enzyme here. One is carbonic anhydrase enzyme. It is catalyzed the carbonic acid and converted the carbonic acid to water and carbon dioxide. The next enzyme is sucrase enzyme. It is used to hydrolysis the sucrose into fructose and glucose. The next third enzyme is lactase. This enzyme hydrolyzes lactose into glucose and galactose. Usually reactant, it gives the transition state. After the transition state, it will form the product. Here the enzyme, when the enzyme it is reacted with the substrate, it reduces the transition state by reducing the activation energy so that it stabilizes the transition state. In this state we get the enzyme substrate complex. Afterward the enzyme will separate it and produce the product. From that, when without the enzyme means we get the product in a slow rate, that is in slowly the product will form. When the enzyme is added means the product will form very fastly, because it reduces the activation energy so that the transition state is stabilized. Here one of the model, the mechanism of enzyme action is given as a model. What model is given means lock and key model. You consider the substrate as a key. What is substrate means? The main reactant only called as a substrate. It is in the form of key, and enzyme you consider as a lock. When the key correctly fit to the lock means the reaction will occur. Otherwise the key it is not correctly fit with the lock, the reaction not occur. It is the mechanism of enzyme action. So far we studied carbohydrate, protein. Now we are going to study the lipids. Lipo means in Greek word fat. The fat will provide 2-3 fold higher energy when compared to the carbohydrate protein. Carbohydrate protein also giving that energy, but when compared to them, the fat, that is lipid, they provided more energy than carbohydrate and protein. These lipids are organic molecule. Usually organic molecules, they do not dissolve in water. They are dissolved in organic solvent like chloroform, methanol. Then the lipids are principal component of cell membrane, one of the point. The second point is it is a energy source for the living system, because it provide more energy when compared to the carbohydrate and protein. It gives more energy, so that we can say that lipid is a energy source for the living system. Lipids, based on the structure, they classified as simple lipids, compound lipids, and derived lipids. The simple lipids, they are two types. One is fats, another one is wax. Fat is the ester. That ester we get from the long chain acid and glycerol, so that we get the triglyceride. That is fat. The next one, wax. It is also a ester, but these ester we get from the fatty acid with long chain monohydric alcohol, for example beeswax. Now compound lipid, that compound lipid, it is the simple fatty acid when it is added with the glycerol and phosphorus or sugar or protein. We get different type of compound lipid. The first one, phospholipid. When the phosphorus is added means we get the phospholipid. The phospholipids contain phosphoester linkage. The second one, glycolipid. The glycolipid means it contain a sugar molecule with a simple fatty acid and as well as glycerol. And lipoprotein, it is a lipid with the protein molecule. When it is added with the simple fatty acid and then glycerol, we get one type of ester, that is called as lipoproteins. Lipids are primary component of cell membrane, and also they are necessary for the structural integrity of the cell, and they give more energy, so that the main function of triglyceride in animals are to reserve the energy. They yield more energy than when compared to the carbohydrate and the protein, and they are the protective coating in the aquatic organism. In integral organ also, it is protected with the help of lipids, and lipids help in the absorption and also transport of fat soluble vitamins, and they activate the enzymes like lipase, and they act as the emulsifier in fat metabolism.